So forward-looking statement for your review. So I won't really, great intro already by the previous speaker. So really not going to get into what iPSCs are, but they're amazing cells that are held in a petri dish that can give rise to all different cell types in the body. And you can really do many different therapeutic applications, from understanding developmental biology to drug discovery to cell therapy. And this slide is just to show that our scientific founders are pioneers in the field. And so we've been fortunate enough to be working with them for the past 10 years, and they've been doing this for much longer than that. And so it's really given us the ability to access really foundational science. And so one of the things we really focused on was single cell iPSC culture. And so what that really gave us the ability was to have a clonal product that has a unique ability to have unlimited self-renewal. So that's awesome. You can always continue culturing these cells. They don't get exhausted if you keep them happy. As well, as we've heard, you can have now the potential to make any of the 200 cell types in the body. And so we focused on that with the mindset of basically engineering the cells at the single cell level to really have a uniform composition of cell product. Now you can think about creating master cell banks for cellular therapy, and now you can do extensive characterization of that master cell bank before you release your product. And so now you have a renewable clonal cell line that is homogeneous in its product. And so this is a cartoon of what we exactly do at Fate Therapeutics. Basically, we take the population of iPSCs, engineer that population, and in a library manner, screen for attributes that we want, and also screen out attributes that we don't want. So when it comes to engineering, look for on-target engineering and off-target effects to eliminate, but also look for the clonal propensity of that particular clone to become the cell type of choice, NK or T cells. And then when that cell becomes an NK or T cell, look at the functional modality that we've introduced and looked at the feature. For example, whether it's targeted therapy or some sort of combinational strategy, see whether that clone that gave you the best NK or T cell also gives you the best function. So when you do all that at the library level of hundreds of clones, then you get to create a truly master cell bank that you've been really investigated and compared to other clonal populations. And so we end up with a master cell line. And I really will focus on how single cell engineering is critical to create a master cell line. The second part of that is, and we heard earlier, is our ability to differentiate the iPSCs to a hematopoietic population. We focus on creating definitive hematopoietic cells that are cultured as monolayer, so you really get to scale the numbers during the differentiation process. And by doing that, we're able to now make T and NK cells. And so I'll talk about how we are able to make large number of T and NK cells. As my previous presentations discussed, cell number is important for therapeutic application, and I'll talk a little about that as well. And so now you have a platform where you can eliminate the stochastic variability that's associated with population engineering, and also be able to create an unprecedented scalable process where you can now create a product in a cost-effective manner. And I'll talk a lot about that as well. And so under a microscope, when you drop a single cell into a 96-well plate, you can see that you have basically a single cell there post-sort. That population begins to divide, 2 cells, 4 cells, and so on. And because we have a small molecule culture, we are able to keep those cells happy in a feeder-free environment and expand them in a uniform manner as iPSCs and maintain genomic stability, as you can see there. And so by doing this, now you can engineer all these cool things in and out that people are discovering through their great work. So for example, here, a cassette knock-in, we took 3 genes and targeted a homology arm, or using homology arms, targeted the AAVS1 safe harbor locus. As you can see here, 3 genes were put into an iPSC population that does not have GFP, but is pure for iPSC based on the flow. And in the same manner, we wanted to knock things out. So for example, we targeted beta-2 microglobulin in an iPSC population. iPSCs are generally HLA class 1 positive, so when you engineer them and knock out B2M, you should have HLA class 1 null cells. And so when we did that, you can see through single cell selection, now we can have a pure population of iPSCs that now have 3 genes targeted to a specific locus as a knock-in example. As a knockout example, you could target beta-2 microglobulin at the single cell level, look for cells that have biallelic disruption of B2M, and now have a B2M null cell that is also class 1 null. So really give you the ability to knock things in and out. So now you create a platform that is very unique when it comes to cell therapy. Although a lot of advancements have been made, cell therapy today is engineered at the population level. And when you think about nucleus-mediated engineering, now you have a whole slew of different cell products that are in your population. Things that are not edited, things that are edited, and things that are edited but have also off-target effect. As you can see there, many groups have really taken this to the next level. 10 years ago, you weren't even thinking about efficient engineering, but today you get very efficient engineering, but still at the final stage, you have a heterogeneous population, simply because you did a population engineering you did engineering at the population level. At Fate, because we start from a single cell and we screen for that, as I mentioned earlier, you have a pure population. And now you really get to do really cool things. For example, after you have screened your product, so here, HNCD16 I'll talk a little more about in the product pipeline stage of the presentation, but we basically took a novel form of CD16 Fc receptor and transduced iPSCs, as you can see in the red dots. As you transduce populations, you get a whole variety of gene expression. And so, as you can see there by red dots, based on the X scale, you have a very differential expression level of the HNCD16 and the iPSCs. We dropped those little red dots into 96-well plates and then screened the population for gene expression. For example, you can see that in the middle, you have a whole slew of gene expression differential of HNCD16 from individual clones. What that has to do with copy number, that has to do with integration site, and that has to result in some sort of function. And so we test all that. We look for the clones that have been integrated in benign regions of the DNA. We look for copy numbers that are preferential, and we look for performance that is ideal. And so basically, after going through all that, we ended up with the purple clone, for example, and carried that forward. That is our master cell bank. That is what we filed the IND with. And we have really in-depth understanding of that population. And so every time we make the NK product from that clone, we know exactly the outcome. We know the differentiation efficiencies, we know the integration site, and we know the performance. In addition to that, amazing, as I mentioned earlier, publications are coming out here. Michelle Satterline, in a very clever and creative way, showed that when you put CAR into the track locus, you get enhanced CAR T biology. If you did that at the population level, go after biolytic disruption of TCR, in-frame integration of CAR into the track locus, you will get heterogeneity. So what I show on the left-hand side is something that basically is population-mediated and looks great for today's standard, looks great for 10 years ago, but it's still a population that is heterogeneous. On the other front, if you did this at the iPSC level, you end up with a pure product at the end that has CAR expression without TCR expression. So really, you can use iPSCs to create very uniform and defined products. And so now this really introduces a process that is more closer to what is pharmaceutical-grade product development. So as we know, autologous CAR T will win the Nobel Prize in the next few years. I'll bet my coat on it. But there are things that need to be improved. And toward that improvement is allogeneic strategies, where the healthy donor is used. Multiple doses are made because you expand the product, but still the challenges remain. It's still population-mediated engineering. It's still challenging to scale, and it's very donor-dependent. When you have that master cell bank, and you can access that master cell bank every single time for every manufacturing campaign, your product now becomes cost-effective and homogeneous. And so it really gives you the unique ability to be more like monoclonal antibody production, where a single CHO cell line gives rise to the entire product for that particular therapeutic antibody. Second part of that manufacturing process is the ability to reproducibly make the product of choice. As we heard earlier, hematopoietic differentiation is challenging, and it does not scale always because you use an embryoid body intermediate. And so what we did was we basically came up with a strategy where with small molecules and cytokines in a stage-specific manner, in a monolayer fashion, we go from an iPSC to a CD34 positive population in 10 days. Continuing that differentiation process that's stage-specific, we can make NK cells. As you can see, the CD34s go through a hematopoietic transition and start giving rise to CD56 positive cells. In a similar manner, we can shift and go toward T cells and start making CD8s that are alpha beta. This process scales because you are expanding at each stage of the differentiation process. You expand at the iPSC stage, you expand at the CD34 stage, progenitor NK stage, and finally the mature T or NK stage. And so there you get about a 10 to the 5, 10 to the 6-fold expansion each time when you expand. And so now you can start off with about 1 million iPSCs or 1E7 iPSCs and end up with 1E11, 1E12 final product. And so each manufacturing campaign can give you a therapeutically relevant number of cells for your phase 1 strategy. And this process scales because the cellular input is not limiting. It scales in a manner that is only volume dependent. So right now, Fate Therapeutics and with our collaborators, we work about 100-liter scale. You could very easily increase the magnitude of output by 1 log by simply using a 1,000-liter scale. So you're limited by the vessel, not by the cellular input. Speaking of our collaborators, our manufacturing collaborators, University of Minnesota is our preferred manufacturing site for NK cells, and MSKCC is our preferred manufacturing site for T cells. And I won't really get into it too much, but we're starting to see that at the phenotypic level and also at the gene expression level, these cells that we're making are very similar to their adult counterparts. They're different because they're made in plastic, but when it comes to effector function and majority of the profiles, they're very similar. And so taking that platform, combining it with the top investigators in their field of CAR T and NK biology, we really started to develop a pipeline that I will talk about now. Our first product is an NK cell, an allogeneic vanilla NK cell. And we're in love with them because NK cells are multifaceted. They have the unique ability to basically secrete a ton of pro-inflammatory cytokines and chemokines. Through their activated inhibitory receptors, they can recognize a multitude of antigens that are found on infected cells or transformed cells while leaving healthy cells alone. They have CD16, which is an Fc receptor that's combined with monoclonal antibodies to enhance the features of the monoclonal antibodies. So majority of the reasons why Herceptin is working and Erbitux is working is because they work with NK cells to eliminate the targeted cancer population. We see that iNKs give you all that and more. The iNKs show that they can activate primary T cells to help primary T cells home and traffic to the site that they're at, and they combine with NK or with primary T cells to exhibit enhanced activity. And so in a simple bar graph, we can show that when combined, iNKs and T cells synergize. So here we did basically an aggregate assay where we take ovarian cancer cell lines, SCOV3, create an aggregate, and then look at the aggregate size and the cells number that's in the aggregate. And so here in red, you can see that SCOV only has about 10,000 cells in an aggregate. When you combine that with about 10,000 iNKs, you can see that there is some effect. And Pembro, which is a PD-1 antagonist, did not really help that process. As we expected, NK cells don't traditionally express PD-1. When you add primary T cells to that aggregate, nothing really happens. When you add Pembro, we see enhanced activity. Only when we combine iNKs and the primary T cells do we see a synergistic activity in eliminating most of the cells. And when we add Pembro to that, we see a clear elimination. And so here, this is kind of, obviously, it's early studies, and it's clinical models need to be further investigated. But in this simple 3D aggregate assay, we can see that there is a synergistic play between iNKs, primary T cells, and Pembro. So that really has led us to push forward with the manufacturing and a clinical trial of allogeneic NK cells to be combined with checkpoint blockade therapy. And so basically this is an example, a snapshot of what our manufacturing capability is at University of Minnesota. So we basically, in a clinical run, we make a product that's frozen and then thawed. Upon thaw, you see that the product is pure for CD45s. It's pure population of hematopoietic cells. It consists mainly of NK cells, as defined by CD56. Viability is high, and the residual of iPSC is not detected. This product goes straight into patients. It is thawed and infused. Preclinical studies show that the product maintains efficacy when it comes to preclinical in vitro and in vivo models. And so we really have now a manufacturing process that gives us hundreds of doses at very low cost to be able to treat patients. We filed the IND and got it cleared back in November. And so this is basically using NK cells to overcome checkpoint blockade resistance. There are many different ways where checkpoint blockade resistance are seen today. As you know, PD-1 inhibitors are changing the way we look at cancer immunotherapy, but there is resistance. Many of the resistance can be overcome by NK cells, whether it's activated T cells that are not in the tumor and can be recruited by NK cells to the tumor site, whether it's the inability of the T cells that are in the tumor site to go after the HLA class 1 low or null cancer cells. Those are ideal targets for NK cells. So basically many of the ways that you can think about resistance can be overcome, and that's why we're very excited that these NK cells can overcome many of those issues. And so the clinical trial is a multi-dose strategy, and again, this is another place where we're very excited about. Usually give NK cells that have about 14 to 21 day in life or have a shelf life in the patient, about 14 to 21 days. By treating the patient on days 1, 8, and 15, you basically now deliver a consistent amount of fresh NK cells to the patient that will create a persistence profile, which we are investigating, which hope to be the case, a persistence profile for 30 days. And so this is the first time that there will be NK cells, adoptive cell therapy NK cells, in the patient for 30 days. And so we will see what that does for the patient. And if the patient shows stable disease, we'll repeat that cycle for a second time. So basically, a 60-day window where NK cells can do their thing. The dose escalation goes up to 1E9 per dose. And so this is about 20% of the NK cells you have in your body. So we will basically, in a 60-day period, will reconstitute the NK compartment into the patients and see what happens. So we're very excited because this is the first clinical trial in the US using iPSC-derived cell therapy, and we're also excited that the first dose has been cleared. Patients have shown no SAEs, and these were patients that were treated 3 times. And all 3 patients show stable disease in monotherapy and have now gone to the second round of treatment. This has now opened up the second dose of 3E8 in monotherapy and the first dose of 1E8 in combination therapy with the checkpoint blockade. So this is very exciting, and we will continue to give you guys updates on this later this year. And as we learn more, we're going to do the kind of really focus on specific patients. And here is a really nice example that patients who have lost their ability to express HLA in their cancer are those that show resistance to checkpoint blockade therapy. And so NK cells are ideal to be ideal combinational agent for this strategy to overcome resistance. The second product that's been cleared by the FDA is, which was in February, is our first engineered iPSC, and to our best of knowledge, is the first in the world to be approved, first iPSC, engineered iPSC product. And so what we really wanted to do here was we want to target an issue that many of us have. So when it comes to therapeutic monoclonal antibody treatment, many of us don't have the high affinity variant. And so about 90% of us don't have that and have inferior antibody response. Those that have, the 10% of us that have the high affinity variant actually respond extremely well, as you can see there with the Herceptin curve. And so in a time where socialism is exciting, we want to bring the 90% to the top 10% and give them the experience. And so that's the product here. Preclinically, it looks really amazing in our hands, where we have combined Rituxan, which is an anti-CD20 antibody against Raji cells in an in vivo model. And when combined with FT516, we see very strong enhanced tumor control, where primary derived NK cells expanded and treated do not exhibit that similar effect. And so this results in an extended survival, as you can see in the bottom. So we're very excited about this. We rarely see Raji cells treated with NK cells show such survival curves. So we're very excited to see that. And this has again led to the second clinical trial, where we will combine in a multi-dose manner again FT516 with Rituxan. And we hope that the high affinity non-cleavable version will augment ADCC, as we see preclinically and improve the patient outcome when it comes to combination with various monoclonal antibodies. And I'll speed up a little bit here as I'll move into more pipeline development. One of the issues that we have seen with monoclonal antibody strategies is things such as anti-CD38 antibody. Although they're effective in eliminating multiple myeloma, they also eliminate highly activated T and NK cells. So just as multiple myeloma expresses CD38, so do NK and T cells when activated. So not only you have an anti-tumor effect, you also end up with fratricide, which reduces the efficacy of drugs such as Darzalex. So what we did, we just simply knocked out CD38. Obviously, we had to investigate how it perturbed developmental biology because it is an endothelial marker, and also it is involved with cell-cell contact. We saw that it did not really alter the ADCC effect. And so here we now have a unique NK cell profile. I hope you appreciate what you see in the flow profile of a pure population of CD16 positive, CD38 negative NK cells, something that has not been shown before. And through iPSC and current engineering strategies, we can now make this cell. And when we make this cell, we actually now avoid fratricide. And when it comes to killing, we see enhanced killing in the second round because there's no longer elimination of NK cells by NK cells. So while in the first round of killing, the first 48 hours, we did not see a differential between the wild type and the CD38 negative NK cell population, targeting the multiple myeloma cell line, MM1S. But in the second round, when we took that same NK population and went into a fresh new killing round, we saw a differential. And that was mainly because there were no more NK cells in the wild type, while the CD38 knockout still maintained enhanced efficacy. So now we're starting to get into multiple engineering strategies, and it's, once again, because I wanted to really focus on single-cell engineering, want to highlight that while the first engineering is highly efficient, and now it's any high school internship can be a guide RNA strategy for getting a 90% plus targeting efficiency. When you bring in a second and third element to edit, you really reduce your efficiency. And again, not every engineered cell is pristine, so it needs to be qualified and quantified. But this is just a reminder that as I'll talk more about complex engineering, keep this in mind. And so the next product we're making is a CAR NK cell. And so the strategy here wasn't to go head-to-head against CAR T, but was to create a product that has multi-targeting potential. So we developed a CAR that was tailor-made for NK biology, so it does not have a 28 zeta costem, it has a 2b4 costem. The transmembrane domain is NKG2D. We coupled that with hnCD16, our non-cleavable high-affinity CD16 Fc receptor, so that we can now come in with a second targeting strategy. And as we know, there's antigen escape in heme malignancies, there is tumor heterogeneity in solid tumors, and so you need a second and tertiary targeting strategies, and hnCD16 gives you that ability to be combined with monoclonal antibodies and start peeling the onion. And so here you can see that an NK cell that is traditionally very hard to engineer, when you engineer at the iPSC level, you can end up with a pure product that has all three modalities. And so when you go into a co-culture assay where it has mixed culture of CD19 positive and negative target cells, you can start seeing that the combination of each modality starts helping you with eliminating the target cell. So in the middle column, no effectors, it was normalized to CD19 negative and positive target cells. Going down the middle column, you add Rituxan to iNK cells, you get a little bit of antibody effect. We see that in vitro culture. When you add the CAR19, you start eliminating the red cells, which are the CD19 positive target cells. When you add the IL-15 receptor fusion to improve persistence, you completely eliminate the red cells. When you add hnCD16 and combine that with Rituxan, you eliminate the green cells as well. And so here is a very unique way of illustrating how you can use a single cell with multiple different agents to eliminate a heterogeneous population. And switching to T cells, obviously working with Michel Satterline, and I believe you heard his presentation yesterday, you're a kid in a candy store, you have access to all these different great ideas, but all these great ideas need engineering strategies to put into the T cell. And so we've been working with Michel to create a product where we make T cells from iPSCs, and those iPSCs are engineered. And as Michelle discussed yesterday, it's a balancing act. It's extremely complicated to make CAR T cells from iPSCs because not only you got to deal with developmental biology, you got to deal with TCR signaling, CAR signaling, and many other factors that alter the course of development. And so working with the very intelligent people in his lab, we've been able to start cracking the secrets of how to make these cells. And I'll just show a couple of snapshots where I'm going to play, see if I could do this. Nope. So here I hope you can appreciate that there are scenarios. So these are all small molecule cytokine modulations in a track-targeted CAR T cell. You can see that there are scenarios where you get minimal number of alpha beta cells, and there are scenarios where you get a ton of alpha beta cells. So you can alter developmental biology, but it's a lot of work, and we're still working on developing this process. At the same time, you can start thinking about making CD4s as well. So I hope you can appreciate there are places where you don't have any CD4s, mostly CD8s, but there are scenarios where you have double positive cells and CD4s as well. So we continue to develop the process. And as I mentioned earlier, working to put a CAR into the track locus with biolytic disruption of TCR expression is extremely complicated. Many are doing it at the pool level. And when we started doing it at the clonal level, it was very interesting to see that when we started with over 500 clones, we actually had quickly funneled down to less than 30 that had both CAR in frame in the TCR locus as well as track edited. So biolytic disruption of TCR, biolytic introduction of CAR, less than 30 cells had that. And so when we went down, we ended up with only 2% of the final population that maintained pluripotency as well as karyotype. And so this funnel effect really shows how heterogeneous a population is, and single cell analysis needs to be done. And if you have an ability to start your product from a single cell, this is the right way to do it. When we do that and when we do it right, a really cool developmental biology occurs. We start from an iPSC that has a CAR in the track locus. As we differentiate to progenitor T cells and double positive T cells, you can see really nice expression of CAR coming up because the track locus is being expressed. And the final product, which is quite reproducible, we end up with a pure population of CAR that has no CD3 expression because TCR is negative as well. So this is really cool, and we're working with Michelle to try to get this into an IND stage by middle of next year. And so just preclinical data shows that in an IP model, you can control the tumor burden, and we're working with various models to show that this can be done IV as well. And finally, I hope I laid the platform that's truly the cornerstone for cancer immunotherapy. Here, basically, the concept of dual targeting continues. You can take whichever CAR you want, plug it into this backbone that has CD38 negative, or it's been biologically disrupted for CD38, has the HNCD16 for a universal secondary engager, and has IL-15 receptor fusion for improved persistence. And so when things come across our way, such as an enhanced BCMA binder, we can simply put that in and now have a dual targeting strategy where we can use the CAR BCMA to target BCMA positive multiple myeloma cells, as well as use the DARA to target the BCMA negative CD38 positive cells. So start really going after the heterogeneity that comes with the tumor population. And so finally, I hope I've shown that cell therapy is really progressing. It used to be basically patient or process-centric, where every single time there was a donor that had to be engineered that resulted in a heterogeneous population. The product was not as homogeneous as desirable, and there was differential in efficacy based on the donor, whether it was a patient or a healthy donor. Using this platform that I just described, hopefully you appreciate that a master cell line can give you a uniform and complete process when it comes to engineering, which results in a homogeneous population. Because it's off the shelf, it's also cost effective, enables multi-dosing strategy, and now it becomes a product-centric process. And so I'll just stop there. Thank all of our collaborators, especially Michelle Satterline, Jeff Miller, Dan Kaufman, Calais Malberg, and all the scientists at Fate Therapeutics, as well as all the other people at Fate, which I shouldn't say scientists, everybody at Fate Therapeutics. Thank you very much.
Questions, please come to the microphone.
Very nice talk. A quick question for you. Do you see any impact on the cargo that you're integrating or knocking in on differentiation process across different patients, or how do you control for that?
I thought I followed your question until you said across patients.
Oh, sorry. Okay, so for your clone,
Yes.
I forgot you have a clone. So how do you control for any impact of the cargo on the differentiation process through lineage commitment?
Sure. Definitely engineering attributes will alter development, and so we are screening for those that are benign for the first part, but those that alter, such as knocking out PD-1, we're coming up with different strategies. But definitely, for example, the TCR, knocking out TCR and having to come in with the CAR, having the ability to put in the track locus and have temporal expression was key. We could not have done it without that concept. So whether it's temporal control or get around it by other ways, it's definitely important to consider.
Can you talk maybe about what FDA wants in an IND package from preclinical tox for an engineered iPSC versus, say, a pooled, harvested cell?
Sure. So we started our cancer immunotherapy program by using donor-derived NK cells, and that was pretty straightforward. We basically had, the tox package was minimal. When it came to iPSC, the main focus was iPSC residuals. Is there a reversion? Do you have carryover? So we really had to show that our product was pure and did not have iPSCs. I think one of the reasons why the FDA was very comfortable with our strategy was because we showed that our product was well defined. It was 100% hematopoietic cells. There were no carryover cells of other lineages. Because we used footprint-free reprogramming, there was no chance of reversion, and we did extensive mouse studies with the maximum dose possible to show that there were no tumorigenicity. So there was a lot of focus on iPSCs, maybe unfairly, but there's a lot of focus on iPSCs. So for the NK cells, they're transduced because there's no gene edit. For the CD38, we use CPF1, and then for the TCRs, we use CPF1. So for both, so we use CPF1 for both. But because we do single cell editing, we've also shown that zinc finger talons is really agnostic to the strategy because you sort out what you want.
Hi, very nice talk. I have a question regarding multi-gene engineering.
Sure.
So in your talk, you gave an example where you mentioned you use lentivirus to make the CD16 clones. So have you also tried using non-viral delivery method generating the clones?
So we've either used nuclease or lentivirus. Do you have a particular strategy in mind that you want to discuss?
No, just in general.
Yeah, so in general, to deliver without having to cut the DNA, we use lentivirus. We can use as much as 3 genes in a lentivirus package, so that's fine. And for NK cells, it seems to be perfect. You need a promoter that actually maintains its activity through development, which is not the easiest thing. So the promoter was important. On the flip side, when it came to cutting, we used, as I mentioned, CPF1. We didn't really use transposons or many vectors, many circles, if that's your specific question. But we've used, iPSCs are very amenable to genetic engineering, so I think whatever you want to use, you can use it on them.
Thank you.
Sure. Yeah, sure. So when you make NK cells without any engineering attribute, they follow the same traditional 7 to 14-day biodistribution. So they have a normal biodistribution profile, and they last about 7 to 14 days in an NSG mouse. Obviously, we haven't done humanized mouse models yet. When you add the persistence attributes, we see it up to 90 days. So IL-15 receptor fusion prolongs that. Sure.
Great. Thank you to all the speakers, and that concludes this education session.